I'm going to try and go back to your question of are we ready to edit our genomes? So towards the end of the book, you write, as of this writing, by the time this book is published, I would not be surprised if the first successful targeted genome modification of a human embryo had been achieved. The sentence ends in a laboratory.
Yeah.
But you then continue, the first, quote, post-genomic human might be on his or her way to being born. So I want to ask you to...
By the way, Bernie, I had expected within the first 5 odd years. It was achieved within the first 5 odd weeks of the publication of this book.
Go ahead.
So just to remind us of the pace of the technology, and there's some debate whether there's a massive debate going on whether that first attempt, second attempt, whether they were actually achieved what they had achieved or not. In the scientific field, there's a debate going on whether in fact they really edited the genes of the embryos, that where they were, if you look in through the details, there's some things that don't make entire sense. So there's that debate, and that will continue for a while.
Right. But in terms of children being born with apparently serious attempts to edit the genome, not in the CRISPR-Cas9 very precise editing, but in mitochondrial diseases and mitochondrial replacement therapy. So rather than very precisely going after a single gene, even though it is a single gene, if it's contained in the mitochondria, why not replace the whole mitochondria? And there at least have been reports of 3 children being born, one of whom apparently meets these criteria: extraordinary suffering, an inheritable, devastating childhood neurological syndrome, Lee disease, which is fatal, confidence that the gene equals the disease. In that family, there had been a history of children being born and dying, and the mutation was understood. And I guess the question, I would want to sort of rewrite your question that you posed on your cards, and I hope you're filling out your cards, passing them to the outside aisles. For the mother, she, I think, said, I am ready to edit my genome. And I guess the other question is, as the scientist at the Fertility Center and the biological PhDs, were they ready? Should they have helped her? So could you talk a little about are we ready, the different we's are different actors?
Well, so the answer is, as I said, I personally was rather surprised that the National Academy, this was not the National Academy is not some kind of body that sits, it's not a body that sits in space. It's made of scientists and people who work in ethics, people who work in, physicians. They brought in inputs from patients, from patient advocates. So this was a highly considered decision. In the gene, as I said before the report was released, it's interesting that this triangle, I talk about the triangle, if I can remember what my proposed triangle was.
I'll very close.
It's interesting. Here it is. This is. It says a triangle of considerations: extraordinary suffering, highly penetrant genotypes, and justifiable intervention has thus far constrained our attempts to intervene on humans. As we loosen the boundaries of this triangle by changing the standards for extraordinary suffering or justifiable interventions for, say, we need new biological, cultural, and social precepts to determine which genetic interventions may be permitted or constrained, and the circumstances in which these interventions become safe or permissible. So this is from the book written about 6 to 8 months before the National Academy report. So the answer is, I think that people are struggling through their individual decisions about these questions. For a mother in a family where you have a devastating medical disease, where you have an extraordinary suffering, nearly 100% penetrance, there is no other justifiable intervention. It's just not like this child can have a surgical procedure or a medical procedure that will alleviate this child's suffering. clearly the decision this mother made was to go ahead and change the genetic information of her embryo. Those questions, I think, are going to become even more amplified as we start pushing against the boundaries of what extraordinary suffering is and are. The one thing I want to make very clear is that it is absolutely true, and others have written about it, I've written about it myself, it is absolutely true that gene editing technologies, CRISPR and Cas9 kinds of technologies, have pushed these questions to the frontier. But you can achieve very similar ends by genetic selection anyway. You could choose not to have a child that bears, without any CRISPR, without any gene editing technologies. You could choose to sequence your embryos or potentially your sperm producing cells or your egg producing cells and not have certain kinds of genomes represented in your future generation. So one of the points that I was trying to make is that these technologies really are intersect with each other, and you should really think of them as parts of a whole, just as you think of your child's capacity to master language in part as learning to read, learning to get meaning, and learning to write. But ultimately what happens is the child masters language and then begins to use portions of that mastery in whatever direction he or she wants to do, sometimes reading, sometimes writing, sometimes learning information, going and finding new information, etc. But the technologies that allow us to make deliberate changes in the human genome exist even without CRISPR and Cas9. They will become amplified with gene editing technologies. They will become even further amplified when we make synthetic DNA in those larger versions, but they already exist. You can choose to not have a child in your family. If you had cystic fibrosis in your family, you could choose to not carry a child with a double mutation. You might choose to have a child who's a carrier, but choose to do that.
One of the wonderful things about your book, to me at least, was the geographical sweep. It starts with a wonderful chapter that the setting is India. You then move to Europe with Mendel, Nazi Germany, the UK, America with the Buck story. This is really a sort of a global issue. And I wonder if the answer to your question, are we ready to edit our genomes, has different answers in different countries and cultures, so that in some countries, as in the US, the FDA has put very heavy restrictions on approving the use of these techniques for embryo manipulation. Other countries, I think, will have very different perspectives. The Ukraine, Mexico, for example, probably China. Can I ask you to talk a little bit about the global dimensions?
So India, China, Korea are obviously of great interest and concern here. In fact, the part of the geographic reach of the book, the last picture in the book of all the photographs is of a clinic in a fertility clinic. Actually, not a fertility clinic, it's just a pediatric clinic in China where every single child is a male. So that every single child being a male in this pediatric clinic occurs with no CRISPR, with no deep learning. It occurs with a crude, but a crude eugenic desire to have male children. So you see how powerful even crude technologies, how powerfully even crude technologies could twist and contort human demographics. And so the question of who the we are and whether we changes from one place to the other, and the question that I raise in the book is, and it's an important question, which is to remind ourselves that privatized eugenics can achieve exactly the same ends as state-mandated eugenics. And that's because the desire for the aspirational desire to have perfect children, lies in the locus of the individual. It does not lie. There's some desires that lie in the locus of the state, but this desire lies in the locus of the individual and is ancient. So it's not as if we were to say to ourselves, oh, let's remove the malignant state actor from all of this and move the technology to the we, that we would get over the problem. The problem is in the we. The problem lies in the we. Who are the we? And what is the we allowed to do? What is the we not allowed to do? So the we, you're absolutely right, is a crucial point. But it's just a reminder that by getting to the we don't escape the question. The democratizing, privatizing, equalizing information does not solve the problem. I use the word equalizing, of course, thoughtfully, because there is, of course, an enormous possibility that only the rich will have access to the technologies that allow them to sequence their unborn children, where the poor will not. What would happen in a world where genetic diseases, or any diseases, or diseases linked to genes, were progressively segregated to the poor, but were not segregated to the rich? What if chromosomal diseases started only happening in the poor parts of a society? There was not just income inequality, but genomic inequality across. But these are obviously real possibilities, and so the we becomes extraordinarily important, but the we doesn't solve the problem.
Okay. I know your lab is actually working on genetic modifications for use in therapy of cancer and leukemia. And there's some questions asking you to talk about the CAR T modifications of T cells. So these are treatments for leukemia, for example, where modifications are made to help the body reject the cancer cells. They're not inherited to the next generation because they're. Is this permissible in ways that don't give us the kind of pauses that germline modification, where the modifications?
Yeah, they're simpler. So our laboratory, because we work on leukemia, we've defined some of the leukemic genes. We made a very simple. It's a very simple idea. I can now disclose it because we now own the patent on it. So the idea is very simple, but I think it has some important ethical and other consequences. The idea is that for a long time, as you know, we were, as a society, as a research community, we were looking for tumor antigens that were specific to a cancer cell that were not present in normal cells, so that we could use those antigens as tools to target, to recognize, to kill tumors. And in the leukemias that I study, in myeloid leukemias, which are very deadly, they're very hard to find. In fact, I have argued, and there's an argument about this, they probably don't even exist. So virtually every antigen, every sign, every flag that we found in a leukemia cell is also present in normal stem cells. So when you go to kill the leukemia using immunological therapy, you also kill the normal stem cells. And this was a big crisis because you cannot deliver leukemia-specific, cancer-specific therapy like you can for some other cancers. So for a long time, we just sat with this idea. We didn't know what to do with it. We scratched our heads about it. How do you make an immune cell go and kill the cancer but not kill the normal cell? Until, of course, CRISPR came along. And we suddenly realized, well, what if you just took the normal cell and you deleted the antigen from the normal cell? So you basically manufactured a tumor antigen. You forced your body to create an antigen only in the tumor cell. And you can do this because of some peculiarities of stem cell biology which wouldn't otherwise be true, but you can do it quite generally. Just to give you an absurd example of this, what if you were to direct an antigen response, an immune response against your liver cancer, but synthesize an artificial liver where that tumor, that antigen was deleted so that it wouldn't be a target? So we scratched our heads about this a little bit more, and using CRISPR began to find out actually it's very doable, it's very feasible to do this. You can actually delete antigens at will. The ethical questions there are kind of enormous, which is who owns an antigen? your body makes it. When you delete it using CRISPR, there are particular tricks of technology. Those tricks of technology are truly synthetic. By truly synthetic, I mean they are not things that are found in nature. You have to make them from scratch. So in that case, does a biomedical researcher own it because he or she made it from scratch? How do you deploy this in patients? What are the safety considerations, et cetera, et cetera? So there's another, even though it doesn't address some of the human genomic considerations, there are a whole nest of questions there which we are now in the thick of, but hopefully we'll get some guidance from about.
Let me try and ask a follow-on question. So the goal of the research your lab is doing to ultimately get therapies that are FDA approved and can be used in patients to treat, perhaps even cure diseases. That process of going from the discovery in your lab to an FDA approved product that can be marketed, prescribed by physicians is a long, difficult, complicated process. It's costly, it takes time. The patent clock is running while it's being developed. To develop for a biotech company to develop that product, this extremely promising product, they need to make it a financially feasible proposition, which means raising money from investors, having the prospect of selling the therapy at a price that recoups some of the investment. So there, in terms of who owns it, if the company doesn't have a clear field to saying, we're going to be able to have an exclusive patent right to develop the disease. And so, but then where does that leave the person from whose cells the technique was either discovered, or if is it an actual patient-centered therapy that each patient gets a different therapy, or they're going to be?
So it depends on what, are you asking Henrietta Lacks' question? I mean, there are two kinds of questions here. Right. So there's a Henrietta Lacks kind of question. It's an important kind of question. So one of the things that's interesting is that that there are technologies that are extraordinarily dependent on individual patient samples. CRISPR doesn't happen to be one of them. For the most part, it doesn't happen to be one of them. Gene editing doesn't happen to be one of them. There are others that are. And you're absolutely right. In those arenas, we've rediscovered, perhaps a little too late, but we've rediscovered that we haven't been given any credit whatsoever to the original samples. The particular technologies we're talking about happen to be relatively independent of all of this. But there is a whole arena, and I would recommend, obviously, I'm sure everyone's read Becca Skloot's book about how to understand giving back material rights to technologies that emerge from physical objects, cells, etc., that emerge from individual patients' bodies. That's a question that really arises out of, really a question that arises when you manipulate individual cells to produce things that are very particular to those individual things, antibodies, for instance, but this doesn't happen to use them.
So a question about the idea of extraordinary suffering. So editing our genome to prevent or treat or cure a disease that causes extraordinary suffering seems to be at one end of a spectrum. And then there's editing to enhance positive traits that fall within a normal statistical range, but trying to push more towards the extreme. Help us think through that.
Yeah, so that gets into, that's really the question. And the reason that's the question is that if you think about it, prenatal genetic testing and selective implantation of embryos is a subtractive technique. So it's the minus sign. And by minus sign, I mean you can take information that's already present and you can decide not to implant certain embryos. You can subtract away information that's already present. But there's no plus sign there. In other words, you can't add to information that mom and, or sperm and egg, I should say, have brought into an embryo. There's no way you can add information to it. There's only, you have what you have. You can select some variants, but you can subtract. The one thing that gene editing, and particularly CRISPR or these techniques allow you to do, is they allow you to add, which no amount of prenatal genetic testing and selective implantation will allow you to do. CRISPR or gene editing techniques allow you to actually potentially add information. It's complicated. It's not clear whether we can do it easily. So that's the one crucial difference. So gene editing, the real test of gene editing, I think, will ride on questions of enhancement and addition of information. Right now, the number of genetic loci, the number of variants where we definitively know that adding information is going to be therapeutic or beneficial are very few. My suspicion is that we will find more. My suspicion is that we will find more and more. And my suspicion is that we will begin to tamper with them more and more. I'll give you one example, again, going back to height. I don't care very much about height, by the way. I'm just using this as an example of what could and what the concerns are. So if you look back at that curve that I just showed you, it turns out that about 50, 60, 70, 80, 100 determinants that have small range effects on human height. That can be relatively comforting to some people like me, because you say that the number of the variation is so great that it's a little unlikely. We can predict, so I can tell you, you can sequence here, you can send your child's genome to somewhere or the other, they can sequence it, and you'll get some information that's spit out and says this child will be this tall. I would say that it's a little bit complicated to act on that information. But it's also true, what was surprisingly true, which we didn't know before, is that there are some alleles that we now know have disproportionate effects on height. So with some small genetic variants, they're rare in human populations. And this is going to be true for other diseases too, other traits too. They're rare in human population, but some of them might govern 1.5 centimeters, 2 centimeters, 3 centimeters of height. How do we know this? Well, we know in cases of severe trade-off, so Marfan syndrome, right? You have a severe trade-off, you get terrifying diseases. But on the other hand, not but on the other hand, and on the other hand, you also get 3, 4 centimeters of height. So those are going to become really interesting, important nodes, because you could say to yourself, now I can really change 1 thing, 2 things, 3 things, and get my 2 centimeters, 3 centimeters. The big question is, can you do that without compromising fundamental pieces of human physiology? In Marfan's, we know it's a terrible compromise. No one would take that compromise. But maybe there are other things. There are genetic variants where a switch from one variant to another variant gets you 2.5 centimeters. The question is, will we accept enhancing those? Will we accept enhancing them when all the other determinants are pushing in the other direction? In other words, someone has familial genetic propensity for short stature. Should we say to those people, you have all this other genetic information where the chances are there's no way I can use genetic selection because of the number of complexities, but I will, but by using this a genetic variant in a very dominant, that has a dominant influence, I will now override these with gene editing. I think that's really interesting and complex universe. People don't think about it because, again, we're still we're still focused on the very base level of this conversation. We're just ascending to deep learning. We're just ascending to understanding how rare variants can influence features more deeply than we had previously anticipated. Okay. So that's what, if you leave this room, that's one of the conversations you need to be having with yourself, is that it is not just gene editing. I'm repeating the point that I just made. It's not gene editing that's the real crux here. It's what happens afterwards. The layer of technologies, just like a child learns, it's the layer of technologies that interposes between gene editing and the next step, finding rare gene variants using deep learning and other techniques, computational techniques, that will reveal rare variants of genes where you have disproportional influence on a feature. Using deep learning to isolate features of human behavior traits, biology that were otherwise imperceptible to our standard computational. That next layer is a make or break layer.
In drawing this evening to a close, I want to help the audience see the future. And
I'd like to see it too.
well, you can see it on PBS next year. There's a Ken Burns documentary
On the gene.
based on the gene. Francis Collins, director of the NIH, will play a prominent role along with Dr. Mukherjee, so it's something for us to look forward to. This is part of a trilogy. The Emperor of All Maladies, your biography of cancer, is volume 1. This is volume 2, and you will be writing a volume 3 on immunology in the future, so we should look for that. We should encourage you to do that.
So the way that they're connected, Bernie, so it's the altered self, the self, and the non-self, the foreign self. So that's how the three are connected.
Okay. So please join me in thanking Dr. Mukherjee.
And thanks to the foundation.
Yeah.